Well, a big welcome again. I know it's been a 2-month break and a bit belated Happy New Year. I know in these troubling times at the moment, it's still very difficult for everybody concerned. But hopefully this might give a bit of light relief on a Thursday evening. And this is, I think, our 12th or 13th webinar that we've done live. So please bear with us. We are, if anything, we're reliant on all the technical know-how that's out there, and Rachel's behind the scenes doing it. So a big welcome to you, and a big welcome to Viagen Pets and Equine. They're going to be joining us tonight. So a big welcome to you all out there in the States. It's going to be we are actually going to be taking most of this all around the States and in Canada. So we're right across the pond, thousands of miles away, and hopefully that you'll see an amazing night tonight. I'm going to go through what we're going to do. So you're very welcome. We want you to ask questions. We've got Faye in the background, and she's going to be any questions you want during this webinar. By all means, ask Faye. And obviously this webinar is all about sort of preservation and cloning tonight. So I think it's going to be one of the most fascinating ones we've done so far, with the sort of most up-to-date technologies that are already out there, and how we can clone our animals, preserve them, and also use these technologies to actually hopefully save species from going extinct. So I think it's absolutely going to be fascinating. I myself are wrapped up in this. I can't wait to hear and listen to what they're doing out in Viagen as well. So Viagen, they're very much be the sponsors here tonight. It's Viagen Pets and Equine. It is a worldwide leader in cloning and animals that we love. They provide the optimum hope for DNA storage of obviously cats, dogs, and horses. And their teams are dedicated to providing outstanding quality and animal care. Viagen Pets and Equine are dedicated to conservation, and through their partnership at San Diego Zoo to revive and restore. And I find this thing, part of it, absolutely amazing what we can actually do with the new technologies that are out there. Tonight, we're actually giving what we're giving away is a free genetic preservation of your domestic animal of your choice, a cat, a dog, or an equine. So we can freeze that down if you do win it, if you want, totally free of charge, just in the future, if ever you wanted to use that. And that's offered through Gemini Genetics. So what's on tonight? Tonight, we're going to talk about genetic preservation and obviously animal cloning. We're going to be asking questions. What is genetic preservation and what's it all about? How does this process go? We're going to be really looking behind the scenes out in America. Some of this footage, I think is very rarely seen. So it's great to be asking those questions that you want to see. We're going to access the technologies via Gemini Genetics and Viagen Equine, Pets and Equine, and exclusively look at some of the things that go on here and how the process actually works. So we're actually going to, at the end of it, we'll actually show you how Lucy's going to explain exactly how this process works in this lab as well. And then at the end of the night, we're going to go upstairs and then do a short presentation about other technologies like sexing semen, epididymal sperm extraction, other preservation technologies. Just while you're on, a note for your diary on the 11th of February, it's all going to be about preparing your stallion for the breeding season. So it's going to be a live webinar again. And any of you out there breeding animals, obviously stallions mainly, we can actually show how do you best prepare your stallion for the busy stud season? How is to get the best fertility results? How are we going to get those best optimum fertility results? So that's not one to be missed. And Rachel will be sending a memo out after. Now, tonight is totally free of charge, as you know, all of our webinars are. But this one's a real personal one because tonight we're going to be raising money. We always do a bit for charity. And we just set up a charity called Nature Safe. And every single penny that you donate tonight will go towards preserving animals. It's one of the largest endangered living charity tissue banks in Europe. And we're basically preserving cells and gametes of tissue samples really to revive and restore for future generations. As we know, this world, the declining and all these animals are going extinct. This is one way we can do it. So a simple £5 donation, if half of you donated, by the way, we've got 1,100 people tuning in tonight, which I think is absolutely incredible. One of our biggest audiences. Last one we did over 52 different countries. So with huge welcome to everybody all over the world. We really are so pleased that you've actually joined it, and I really mean it from my heart. I so appreciate you tuning in and watching it. So at the end of this, you will get an email tomorrow or the next day saying, please, can you donate? It's entirely up to you, but if you can donate £5 or £10, every single penny goes to saving animals from extinction through Nature's Safe. So £5 donation will actually save about 50, if every other person donates, about 50 different endangered species. So, and also a big thank you for British Breeding. Without them and providing the platform tonight, we couldn't do all this. So thank you, Rachel. Thank you, Jane, and thank you, Eva, for the British Breeding to supply the platform. So, right, let's crack on. And the first thing we're going to talk about is a little bit about Gemini Genetics. And Gemini Genetics is part of Stallion AI services that are here. And it's myself and Lucy that you'll see in a minute. It was established in 2018. And it's one of the first UK animal living tissue banks that was set up back then. And it was mainly set up, obviously, for preserving rare equine preservation. But obviously, we do a lot, obviously, in the pet and cat and dog market as well. And now we've, for the last few years, we've teamed up with Viagen, and they have been instrumental in helping us set up this. We're their sort of European hub, you can, so to speak. So all the cloning's done over in the States, but they're going to talk about that. But I've got to introduce you now. Lucy Morgan's one of our lab technicians. She's not just a lab technician, she's actually the manager, and she's a bit too maybe shy to it, but she's the brains behind a lot of this, not me. And she's runs Gemini Genetics for you. So if you can just show us, Lucy, a little bit around this lab, we'll come back to it later, and then we're going to go straight over to America in a minute. So, so just explain what you're doing here, Lucy.
Yeah, that's fine. So here we do the genetic preservation stage, which is where we get the animal tissue and we preserve down its genetics so that those genetics aren't going to degrade. When an animal passes away, its body tissues and DNA genetic material within it would normally degrade as the animal does so, and what we do here is to prevent that degradation. So quick look around the lab. We have the biological safety cabinet, which is where I work. It ensures that we have a sterile environment for processing the tissue samples. One of the main risks to sample viability is contamination from environmental pathogens, so we have to make sure that everything we do in the lab is really very clean and sterile. And then behind Tullis over here, we have our CO2 incubator, which is where we're starting the next stage of our work, which is cell culture. So this is where we, after preserving the genetics, we ask the cells to regenerate, and the purpose of this is to have enough genetic material in order to regenerate a living animal should you wish to do so. So we have a couple of samples in there, as you see, but Viagen Pets and Equine will be showing you more about that stage later on. And then behind over here, we have our DIC inverted microscope, and this basically enables us to look inside the cell culture dish and to monitor the cell growth within. So on the screen there, you've got some cells that are currently growing in this dish here as part of our establishment of this stage.
Come back to that a bit later. Okay.
Yeah, and we'll be coming back to all that processing a bit later on.
Great, right. Well, that was just a very quick intro into the lab, and so now we're going to head all the way over the side of the pond. It's a bit better for them because it's in the middle of the day for them. So for all us lot, I bet all you lot at home have got a nice pint of beer. I bet Shirley I bet you've got a beer if no one has. And I know we've got some good people tuning in. Shirley, Terry, I said you were going to tune in tonight. She's actually one of the ladies that really got me involved with this in the first place. Yeah, so appreciate everybody listening in tonight. So I really wanted to introduce Blake Russell. He is the CEO of Viagen, and Blake's going to tell us a little bit about how he started. So Blake, can you hear me okay?
Yeah, I can hear you great, Tullis. Can you guys hear me fine?
Yeah, we're coming through loud and clear. It's like you're literally next door to us. So if this isn't social distancing, I don't know what is between you and me. So,
yeah, perfect.
So, you know, Blake, we met about, I think it's 3 years ago actually now in May. I came over for a rare breeds meeting and you said, hey, Tullis, I remember you drove 4 hours. I'd never met you before, and you drove 4 hours and you came and see me, and I think you said, oh, we've got this cloning company that goes on. Would you like to be a part of it? But how long have you been doing this for now, Blake?
Yeah, Tullis, first off, I would like to echo your sentiments to the group. Thanks to everyone for participating. The whole team here at Viagen Pets and Equine is really excited to tell you guys more about what we do and why we do it and how we do it. So thank you guys for tuning in. And Tullis, Lucy, and the rest of the group at Gemini, hey, thank you guys for allowing us to be a part of this. So, we've been around, Tullis, for about 20 years now. Viagen really formed on the back of the successful cloning of Dolly the sheep at the Roslin Institute, and we're the exclusive global licensee of the cloning technology, the somatic cell nuclear transfer technology that came out of that. And so as the company was formed, it really came about putting together all of the intellectual property necessary to be able to successfully clone. And from there, really, we began in the livestock and horse space. So we've been cloning horses, cattle, goats, pigs, sheep, deer, those sorts of things now for nearly 2 decades. And then about 5 years ago, decided that there were really different segments in this space that needed to be served differently. And so the group that's speaking to you today is Viagen Pets and Equine. So we focus on horses, dogs, and cats, and then conservation species. So you'll hear more about some of the endangered animal work that we're doing. And that's really an exciting part of our business. And as those of you out there are aware, working in the horse and the companion animal space is quite a different space than the livestock market. So the team that you'll be hearing from today, and again, I've got my co-host here, who's a 16-year-old clone of Royal Blue Boone, a very famous quarter horse here in Texas. In fact, when people drive by my place, they honk and wave at her, and they don't even notice that I'm here. But anyway, moving on, the team that we have here have all been with Viagen for well over a decade and have been a key part of us growing and developing this business. So again, from all of our team, thank you for letting us be a part. You're going to hear about 20 years of our experience building and developing this business, and here's something about our dreams and plans for what lies ahead.
No, well, that's great. And is it a mare behind you? Is it?
Yeah, so this is a 16-year-old cloned mare of a very famous quarter horse that was about 25 when she was biopsied. So one of the questions that we often get, right, is as an animal ages, are you still successful taking a biopsy from an older animal? Well, this lovely lady behind me was cloned from a late in life famous American quarter horse. And again, this gal here is 16, has had numerous offspring successfully. And as you could tell, if I had a wide angle lens, she's about 280 days pregnant now. So she is a terrific producer and has produced some amazing offspring. She's really the centerpiece of our ranch here.
Yeah, she looks like she's stealing the limelight off you a little bit there, Blake, I'm afraid.
Yeah, she's far more lovely to look at than me.
So, and have you seen, you've been doing this 20 years now, so there must have been a huge, maybe we speak later to Sanchez later, but have you seen a huge involvement of cloning and tissue banking? You must, because technology moves at such a rate, doesn't it, nowadays? But 20 years ago, you must be in a totally different place than you are now.
It's exactly right, Tullis. If you think about the tens and literally nearly $100 million that we've invested in research over that 20 years, it has placed us in a very different spot today versus what people think of when they think about the cloning of Dolly the sheep and the literally hundreds and hundreds of attempts that it took to produce her. And we've continued to get better every year. So not only are we more efficient, which then impacts our ability to get our products to clients at even a more affordable price. But, we continue to learn more about all the husbandry aspects of what we do as well. And so our animals, we continue to remain dedicated to our animals being healthy and happy and long lives. And certainly over that 20 years, our team has developed a very unique set of skills to be able to do that effectively for clients around the world.
Yeah, well, I'm really looking forward now to looking at more technology. And I think we're coming back to you obviously at the end, but I think we're moving on to. I'm sorry, everybody knows how bad I am at names, so I'm just putting it out there. But get your names wrong, Mullane.
Yeah,
Mullane.
I think the next step for us, Tullis, would be to hand this over to our client service leader, and that's Mullane Rodriguez. And Mullane has been doing this for a very long time. So not only does she have a lot of direct experience dealing with clients and across all the species, but she also gets heavily involved in the transport, the regulatory pieces, answering all of the questions. As the people around the world that are listening to this today have questions come up, I have no doubt that many of those will be directed to Mullane. So I think Mullane is going to, from her home with her cloned cat, is going to walk us through some of the genetic preservation pieces and some of the logistics.
It might be, before we go there, just 2 seconds. Could you just thinking so people know where we're going? So Mullane is from Georgia. Can you just have literally 30 seconds on each where we're going after that? So we're going, who are we going on to now? Sanchez after that.
So currently I'm coming to you from my foaling barn in Gainesville, Texas, which is North Texas, which is really the home of quarter horse country, right? As many people around the world would know. And then we're going to go to Mullane Rodriguez, who's located in Savannah, Georgia. She's client service leader. Then we're going to go to our cell culture laboratory in Austin, Texas. Sanaz will be teaming up there to share with you our cell culture process. And then she's going to hand that over to our chief science officer, Sean Walker, who'll be coming to us from Alberta, Canada. So we're literally going to bounce all around the US and then jump into Canada for a bit as well.
Wow, the modern technology. Yeah. And that's one thing that with the COVID situation out there that's forced us into this, is if there's a tiny little bit positive, it's the connectivity now that we're all used to and can do now. So it gives us an opportunity that we never normally do, be able to look behind the scenes. So that would be great. Okay, so it'll be great to go over and see Mullane. Are you there?
All right. Hi there. Well, hi, everyone. It's great to be with you guys. As Blake mentioned, I'm the client service manager, and I've been here with Viagen for about 12 years. And my role as the client service manager is to really walk the client through the process from start to finish. You know, as we know, this is a complicated subject, lots of questions to be asked. So I'm here to help facilitate that in any way that I can. So behind you, you'll see my partner here. This is Benji. He is a cloned Bengal cat. So he is cloned from an award-winning show cat, and he is about 2.5 years old. So he's a great example of the breed and a great example of a cloned animal. Just looking at him, you'd never know that he's a clone, other than he's just a really gorgeous, great cat.
Yeah, he's been sitting there for about sort of half an hour. It's so peaceful as soon as the cameras came on. I have to say, he is one of the most beautiful cats I've ever seen. But sorry for interrupting. I think that's lovely.
Well, we've been feeding him treats, so that's keeping him very happy. As you can see, he's a big boy, so he likes his treats. So I'm just going to give you sort of a brief overview of how all this process works. Cloning an animal is going to start with genetic preservation. And what genetic preservation is you're really just preserving the genes. So that starts with a biopsy. So the dog, cat, or horse will have a small tissue biopsy taken by the veterinarian, and that tissue sample will come to our lab, and we will grow millions of cells from that tissue sample. And those tissues, those cells, will contain all the complete DNA from that animal, and that's going to be the DNA source when it comes time for cloning. And you'll hear a little bit more about cloning later on in the talk. So that's that first step, is growing cells. We have a biopsy kit that we would ship out, shipped by FedEx. For clients in the States, we ship that overnight. We do have clients worldwide. I see there's people from all over the world joining on this call. That's great. So we've shipped kits to Australia, New Zealand, Japan. So it's really great that we can offer these services throughout the world. You're looking now at our biopsy kit. This has a biopsy punch tool that's used to take the sample. It's a 4-millimeter punch for a dog or cat, and a 6-millimeter punch for a horse. So it's a minimally invasive procedure, a small piece of tissue. This sampling can be done with just a local injection of a lidocaine at the biopsy site. So very easy, quick recovery time for the animal. This kit is going to help keep these tissues cold. So the most important part of sampling are time and temperature. So the tissues have to stay chilled. There's ice packs inside this kit that will keep those tissues the right temperature. And we need to get the samples within 5 days. So for those of you that may be around the UK area, we do utilize Gemini Genetics. So they can process tissue samples there. So we work with them quite frequently for that part of it. So that's going to be the first step, growing cells from these tissue samples. And then we cryopreserve these cells. We can keep these cells stored for decades. So you don't have to move on right into cloning right away. If you're not quite ready for that, you can keep the cells preserved for decades. We're going to talk a little bit later about the Przewalski's horse. That DNA was preserved over 40 years ago. So you can certainly keep these samples stored for years and years and clone at any point down the road, or you could clone right away.
One question that we had before, just about it, what was the one about the cells being stored?
So Lindsay has asked, can length of genetic preservation lessen the quality of the cells when ready to use for future cloning?
So I presume if they're stored for a long period of time, once they're frozen down, is it going to make it worse?
So absolutely not. There's no degradation of these cells over time. And like I mentioned, we had the horse that we've cloned where cells were stored 40 years ago. So we've cloned several dogs whose cells were stored for 17 years. So these cells are essentially viable indefinitely at the cryopreserved state.
And I think it's just great work. I know we're coming on to, I'm always itchy to go on to the rare breed stuff you do, because I find that so fascinating. The work you're doing for the pet side has really led on to that. But again, I think we'll come on to that bit after. Yeah, another question was it?
One question actually come in. Can you freeze the tissue samples, or are they sent chilled?
So we could do both essentially. So there at Gemini Genetics, they can freeze or cryopreserve the tissues there. There's a special technique that's used to cryopreserve the tissues. So they're not just put in a freezer and frozen that way. They're put in a cryoprotectant media to prevent any damage during the freezing process. So then at that point, Gemini Genetics could ship these cryopreserved tissues to our lab in the States, and then we can culture the cells there, or we can keep those tissues cryopreserved.
Yeah, thank you.
Right. Oh, go ahead.
No, carry on. I was going to say, yeah.
So as we talked about these samplings, it's always preferred that the samples be taken while the animal is still alive. That's going to give us the best chance of success. One of the common questions that we get is, can the tissue samples be taken from a deceased pet? Are these samples still viable? And the answer is sometimes. It depends on the situation. So as I mentioned, timing and temperature are crucial. So if the pet or horse has passed away, those samples need to be taken as soon as possible after death. If it's a small animal, the body should be refrigerated, or those tissue samples taken and refrigerated, and then they need to get to us as quickly as possible. So the tissues are viable for about 5 days as long as they're kept chilled at refrigerated conditions. So we've actually cloned lots of animals whose samples were taken after the animal had passed away. It's always best to be proactive and try to get these samples taken while your pet or horse is still alive. It's a very emotional situation when your pet or horse passes away, so it can be a little bit complicated. So you definitely want to try to get those samples taken, preferably while the pet is still alive. And for horses, particularly for horses that are located outside of the United States, there's actually an import permit that is required in order to get those tissue samples into the States. So that permitting process can take about a week or so. So if the horse is deceased, there's really a very slim chance of getting those tissues into the States, getting that permit in time while those tissues are viable. And that's one place where Gemini Genetics can help us out. They can cryopreserve the tissues there while we're getting those permits, and then those tissues can be imported at a later date. So have the samples taken from a living animal if possible, or utilize, we have emergency instructions on our website that explain how to take a sample from a deceased pet, and then we cryopreserve those samples. So now moving on to some frequently asked questions. Of course, especially with pets, the most common question we get is the personality going to be the same? You know, that's why we love our pets, the personality. So, you know, we started cloning pets in 2015, and we really weren't sure what those personalities were going to be like. You know, we had been cloning livestock before that. I know those species have personalities, but the feedback from those clients was more, you know, is their milk production going to be the same? Is their semen production going to be the same? So we didn't get that personality feedback from those species, but now that we've been cloning horses for or pets for over 5 years, we're getting that feedback now. And what we're seeing is sometimes the personalities can be very similar. They may not be, but the feedback from those clients is that when they see that cloned dog or cat, it reminds them of that original animal. It's an essence of that original pet, and they're very happy about that. But some animals do tend to be, the feedback has been that they are very similar to the original. Personality is really a mixture of the environment and genetics. So things like temperament and intelligence, we know that those are highly heritable traits. So we definitely see similarities in those areas. So let's see, I think that's all I've got about now. I'm sure we'll have questions coming up later on.
Yeah.
So I think we'll pass it on to Tullis for a poll question.
Great. Okay. Well, thanks very much for that. There wasn't any video to play then, was there? No. Oh, slideshow was it? Yeah. Oh, can you see? Just a poll question now for everybody. It'd just be quite interesting to find out. We've got about, let's say, over 1,000 people tonight. It'd just be interesting what percentage of you vets. So we're going to put up a thing saying, are you a vet or are you a layman? And so if you just literally reply yes or no to that. And while we're doing that, you're asking that, there's so many questions coming in, which is great because there's interaction. So Faye, is there a question that I can maybe throw out to the floor and see?
Yeah, a few people have asked, is it possible to clone a clone? And what are the registration kind of implications with that?
Yeah, so I don't know who wants to answer that, but I think it's been done that. But can you clone a clone?
Yeah, I'll be happy to answer that. Definitely yes, and it happens all of the time. And we've had clones produced from clones of clones of clones, I think, at this point. So it's happened quite often. From the registry perspective, every registry will deal with this differently, right? So it's difficult for us to make a general comment about registry.
I think we've got someone's child in the background talking there. I don't know whose it is. There's obviously someone's trying to overhear you, I think, Blake.
Okay, well, there's no children here, so that can't be me. So anyway, I think you heard our answer there. Yes, definitely can clone from a clone, and there have been no issues associated with that. And the registry question, maybe we can deal with more specific registries later. Most horse registries are accepting and registering clones today, but there still are several that are not.
Right. Okay. Brilliant. Do you want to do one more?
Yeah, do one more. So, are all animals able or allowed to be cloned? Are there any exceptions?
Well, so again, there are certain things necessary for our process, right? So we need a sample, right? So we can only clone animals of which samples were taken. We need eggs, so there have to be oocytes available from either that species or a species that is similar enough that it will work as a host egg. And then we need surrogates. So in some of the endangered animal cases, for example, if there's not an appropriate number of surrogates available either of that species or a very closely related species, and Dr. Walker will share with you some examples with both the Przewalski horse and the black-footed ferret where we're able to find more abundant surrogates from a similar closely related species. But I think that answers your question. There would be certain mammals maybe that might be protected in certain places and they wouldn't want this kind of effort. But I think in most places this would just be considered a very precise reproductive technology that, if it can help, would certainly be brought to bear.
Brilliant. Have we got the poll answer now? Thank you for that, Blake. So here we have 56% of the people, 44% of people are vets, and 56% of people aren't. So just click that off. So that's just quite an interesting scenario. So now we're moving on to Sanchez. I hopefully have Sanaz. Sorry. Now I think this is we're going through all the different stages, and we're sort of finishing off where the actual clothing takes place. But we're going to see Sanaz now. And we were just, when we were talking about Malaine, I got that bit right, about taking the sample. And obviously with these samples, and you have to give me if I get this wrong, because I'm not the expert for this, you are out there, but that's only one part. The next bit, we've got to replicate these cells and get these cells to survive. Now, I'm amazed what Lucy does in this lab now. It's like going into a greenhouse. She feeds these cells each day and makes them grow. And so we're going over to Sanaz and going into the cell culture map. So you can, are you there, Sanaz?
Yes, can you hear me?
Brilliant, yes,
Okay.
yes.
Okay, so my name is Sanaz Arnivas. I'm the cell culture manager here at Viagen Pets and Equine. This is our cell culture lab here in Austin, Texas. We process equine, canine, and feline samples here in this lab. So once our client has taken samples, their vet has taken samples of their genetic preservation kit that Malaine showed, we receive the kit here. We process dozens of samples weekly, and we process all the different species here in this lab. So as Malaine mentioned, we do prefer to receive skin biopsy samples from live animals and samples that have been kept cold, refrigerator temperature, during the transit and after collection. But we do receive a lot of samples from deceased animals as well. And like she mentioned, temperature, condition of the animal, and the way we receive it upon arrival here in the lab, and the number of days that it has been deceased can all lower chances of success for viable cells. With that being said, we have had many success stories of these kind of unideal compromised situations. So things like maybe the animal was frozen for a small period of time before the collection of the samples, to animals that maybe were buried and then exhumed after a couple days, and then samples sent to us. So with the genetic preservation kit, we include 4 vials. We like to get 4 different skin biopsy samples here, but we do require 2. So you can take up to 4, but at least 2 different skin biopsy samples. So for dogs and cats, it's a 4mm size biopsy sample, and for horses, it's a 6mm size biopsy sample. For dogs and cats, it's a skin sample, usually from the abdomen, the back or shoulder, or the leg areas is where it's collected from. And then horses, it's from the mane, the crest, or underneath the tail is locations where samples are taken from. So Katie here is processing some, yes?
I was just about to say, are you taking it from, those are skin samples. Is there any advancements in technology moving on from blood or any other urine or anything like that?
Semen is one we're getting.
Someone's asking, can you clone a horse from semen? That's
Yeah,
a question.
so technically, if you do get cells, yes. There's nothing in place at the moment, but the future, that is the goal in the future, is to find less invasive ways to be able to collect cells from an animal to be able to culture cells.
And I think we've missed off one video from the last one. Is that right, Faye?
Yes.
So they've just asked to play that slideshow.
Yeah.
Rachel, can you just play that slideshow? I think that was meant to be played on the last with Malaine. You might want to talk over this. So who's this? Oh, this is the one we just seen.
Malaine, can you mention who these animals are?
Yes, yes. So these are some photographs of some cloned animals. That obviously is Benji, my cloned cat right there.
Oh, just the two, was it? Right. Sorry, maybe that didn't work so well. Was there meant to be quite a few pictures there? Was it? Don't worry, we'll go back. We'll go back to Sanaz. We'll crack on with yourself, and you carry on explaining about what you do.
Sure. So as I was saying, the skin biopsy samples are fairly small. So for cats and dogs, they're 4 millimeter, and for horses, they're 6 millimeter in size. So Katie here is processing some samples that we received today, and I just want to show you the size of the biopsy sample. So the first one that you're seeing, these are 4 millimeter in size skin samples. And then the other dish that she's going to show you, these are 6 millimeter in size. So fairly small samples is what we need to be able to preserve viable cells from your animal.
And can I just, one thing that we have sometimes is that it's got to be chilled at the samples, but if they put the animal and freeze the animal down, that's no good for the cells. Is that right?
Correct. Contrary to maybe what you would think, freezing an animal or the samples prior to us receiving them in the lab actually is a negative effect to the cells that are in the tissue. So the goal is to keep the skin biopsy samples cold, refrigerator temperature at all times, and during transit as well into our lab. And then we go through our processes, and eventually we will cryopreserve cells and the initial skin, a little bit of the initial skin sample we receive in the correct cryopreservant that will preserve the samples.
And you have to be ultra clean, don't you, for this? Because obviously, when you preserve it, everything else wants to grow in there as well, I presume. So the sterility is king, I presume, in a lot of this. Is that correct?
Yes. When we receive samples in here, we go through multiple steps to sterilize and clean the samples. We do have some steps during the biopsy procedure where the area should be cleaned, but we don't expect them to be perfect. So we do go through some steps to make sure we get rid of any any contamination that could be on those samples. So we utilize the biosafety cabinets here that Lucy showed in her lab as well to process the samples. And then once they're processed, we use our incubators that are temperature and CO2 controlled. And we place these samples in flasks similar to this into our incubator. These these samples stay here. We observe and monitor them daily for cell growth. They begin to multiply and grow. And once they get to the confluency or the number of cells that we like to see, then we go through the steps to cryopreserve the cells.
How many days does that take from when they come in to being able to freeze those cells down after growing the cell lines, roughly?
Yeah, so there is variation, but from the day we receive the tissue samples in our lab until the day they are stored in their ultimate freezer or cryopreservation storage, it's usually about 2 to 3 weeks. And that can be, some samples might take a week and some samples might take a little longer. It just depends. Every sample, there is some variation, and the size of it does make sometimes a difference too.
Yeah, I see by the clock in the background, it's about 20 to 2 in your country. So we're just holding out here. It's getting very dark. But you must be, yeah. You must find it absolutely fascinating, your job, being at the cutting edge of science. You know, I'm dipping a toe in it over in the UK, and I really feel like we're always trying to push the boundaries here. But you really are pushing those boundaries and helping the pet market. But all this technology that you're learning is obviously going for other species as well. So you must really get a sense of well-being from doing what you're doing.
I enjoy it. If it's a personal question, I've been here over 17 years, and I still love what I do. So The procedure we use is fairly similar, regardless of species, but every sample that we receive in the lab comes with a different story, and it's just very neat to see the reason a lot of times why someone wants to preserve the genetics of their pet or horse, and maybe ultimately clone that animal. So I enjoy it. Yes. So, sorry.
Go on, carry on. Sorry, carry on. Yeah.
So ultimately, for every animal that we get samples for, we freeze at least 1 million cells. So for every animal, at least 1 million cells, a lot of times more than that, but at least 1 million cells is what we freeze. And all our steps that we take in the lab are proprietary methods, and we do optimize them as needed during time. So once the cells have gotten to the point where they are ready to be preserved, and we go through those steps, they get frozen in these cryovials. So these are 2 mil in size cryovials that can be frozen in very cool temperatures. And we store the cells in these vials for the samples, and we place them in our liquid nitrogen dewar storage system. So follow me, I'm gonna go to where we have a storage system to show you. Okay, so let me put some gloves on. So this is a liquid nitrogen dewar. It holds, this one holds over 25,000 vials or samples for different clients. So these liquid nitrogen dewars is what we store our samples in. They are stored in the vapor phase of liquid nitrogen, which is actually a little colder. And just to let you know, it's about negative 196 degrees Celsius. So very cold temperatures that keep them cold and preserved correctly. So in these dewars, in this specific one, we have what we call these racks. So we have about 24 racks in this specific dewar. We have in these racks, these boxes, and these boxes store those same cryovials that I showed you earlier. And each of these boxes holds 81 slots for these vials. So as you can see, these storage dewars hold a lot of samples. We have thousands of clients stored from all around the world in these storage dewars, and we are rapidly growing and expanding.
You look like you've done that. You look like you've done that a few times. There's obviously it looks like there's hundreds of animals in that particular one. You seem very. It looked a pretty cool way of doing it, because obviously with the semen side we're always using straws and you're doing cryovials, but it's all kept in the same way, so to speak, liquid nitrogen, isn't it? Oh, can you hear me?
For that animal, and we've stored them in these liquid nitrogen dewars. So a portion of every animal samples that we preserve down goes to our state-of-the-art biostorage facility partner, Cryoport. So Cryoport is a leader in their technology in this field. They store samples from NASA. They house samples from globally known health providers like MD Anderson. With these multiple locations that we store your samples in, gives us the fullest confidence in our cryopreservation storage system.
Yep.
In addition, we have a multi-layered 24/7 monitoring system in place in our labs, to where we can at all times have eyes on our samples and everything that happens in the labs, and of course our storage dewars. The samples here, once they're in their final destination, they're here indefinitely until the client is ready to maybe move on to the cloning steps.
Great. What percentage of people go through to the cell line stage? Do a lot of people just keep them as a tissue and just keep them as a tissue, or do most people want to put them into cell lines as well?
So most of our clients, I would say more than 95% of our clients do the full genetic preservation. That's because then you have the 100% guarantee that you are able to get viable cells from the skin biopsies. But with that being said, we do have some clients that do our express tissue bank. And what that is the skin biopsy samples that we receive, we freeze down and I cryopreserve it and just preserve the skin tissue. And that gets stored, and maybe at a later time, the client decides that they want to move to the genetic preservation. And in that time, we would thaw the tissue and grow the viable cells there. Our efficiency with thawing skin tissue from an express tissue bank order is very high. So I feel comfortable enough if someone were to preserve tissue and then at a later time thaw that to viable cells. Sometimes with, I think a lot of people, especially if their animal's deceased, this is the samples that they would, they kind of want to know, do I have viable cells or not? If the animal's still alive, you still would have the chance to go back to collect more skin tissue samples if we were not able to be successful at first.
Brilliant. Well, that I think's given us a really, really good overview. Really, really appreciate that. So was there any questions you want?
Loads of questions. A few people have asked about the size of the animal that's produced after the clone. Is it often smaller than the original? And other traits, for instance, you mentioned, Melaine mentioned earlier the temperament, but it's physical attributes as well. How are they different?
So I think Sean can answer that since he's going to get the mic next.
Okay, well, we'll get that one then. Just before we go there, I think they wanted to try and show that video again. Was that right, Faye?
Yes.
So,
yeah.
Rachel, can you just try and play that video? And then I think if Melaine, if you can just talk over it when it's been played.
Okay, great.
Hopefully it'll work this time. Maybe not.
So there's Baxter. He's a cloned dog. This one here, I don't know if we can pause this video, but this is a great example of phenotypic variation. So one of the common questions we get is, is the cloned animal going to look exactly like the original animal? So this is in the middle. This is a long-haired Chihuahua named Bruce Wayne, and he's got 4 clones. And as you can see, the white right down the middle of their eyes is a little bit different in each one. So this is phenotypic variation caused by epigenetics. So you can have slight differences in markings. So that's the most common difference, I would say, that we see in cloned animals. And also, this is a good example of multiple puppies being born. So we do transfer more than one embryo into the surrogate. So much like in vitro fertilization in humans, you could have twins or triplets, or in this case, quadruplets. The cloning is just one flat fee, no matter how many are born, and the client would get all the puppies. So this client certainly has her hands full, but she couldn't be happier. Okay. So these are some of our first cloned kittens. So these are now 5 years old. This was when they were very small. So here there were 2 born in the litter. And they looked identical, exactly alike. Another beautiful cloned puppy. One of our first cloned cats. Lots of beautiful pets that we've cloned, all different breeds. This dog has an amazing story. So this is a clone of Marley. These clients have an interesting story. The wife was pregnant, and they had a dog named Marley, and they were hiking in the woods, and Marley jumped all of a sudden in front of the wife, and it turns out there was a rattlesnake that became very close to biting the wife as she was very pregnant. So he essentially saved her life. They were out really in the middle of nowhere, very far away. So he saved her life. So years later, they cloned Marley and have this beautiful puppy, and couldn't be happier. Beautiful clone foal. As you can see here, markings slightly different. And that's the Przewalski's horse. And I think Sean will talk a little bit more about this one.
Yeah, quite a special one, that one. Yes.
Yeah.
Well, thank you very much for that, Malene. Yeah, I will get it by the end. Now, and certainly not least, I think, we're saving sort of the real science side to the end that a lot of you might be really interested. Well, not the end, we've got a lot more to do. We've got to show you around the lab and everything. Oh, I hope you can hear me. I think we can see Viagen Pets. We're going to hand over to Sean now. Sean, can you hear us?
Yes, I can hear you. Can you hear me okay?
Yeah, brilliant. So it's great to be, you're in Canada now, so we're literally going all over the other side of the world at the moment. And you're at home, I believe, with your clone dog. Is that right?
Yes, so we're here with two clone dogs here, two of our clone dogs. So this would be Baxter. Baxter, he is a 3-year-old toy poodle that basically is a very affectionate and loving animal. And we have a 5-month-old Great Dane. His name is Leo. I will try to see if we can get Leo in here. Hey, Leo. I don't know if, yeah, there. That's Leo. He's not really overly interested in anything today except for his Chewy. And so as you can see, he's quite a large Great Dane there.
But you do all the cloning, so you perform all the techniques, is that right over there? So you actually perform the cloning procedures on all these different species?
So basically our cloning lab for pets is actually in New York. So we have a different group that works in New York. We have a group of 2 individuals that actually work up in Canada that are doing 100% of the horse work.
Right, right. So yeah, so if you can just go through some of the sort of the process and some of the work you're doing, obviously the actual cloning side would be great.
Yeah, so we'll just go through, I'll give you a little bit of background about myself, Viagen, and then we'll go into some of the endangered species work, and then we'll go ahead and hit a little bit about the technology too. So my name is Sean Walker. I'm happy to be here. Thanks for having Viagen Pets and Equine. I am the CSO of Viagen Pets and Equine, which just means that I oversee kind of the general operations from research and production standpoint, in addition to doing the daily horse cloning work. I've been with Viagen for 20 years now. I began my cloning work with PPL Therapeutics. PPL Therapeutics actually had a division in Blacksburg, Virginia. They're the ones that are famous for cloning the sheep. While we were there, we were focusing on work with pigs. We were making xenotransplantation models, so animals for organ transplants. From PPL Therapeutics, I went to Texas A&M. I got my PhD in genetics with a specialty in cloning animals. So while I was finishing my PhD at Texas A&M, I actually got hired at Viagen to oversee their cloning operations. A little bit about Viagen Pets and Equine. It actually started 20 years ago, as Blake mentioned. We actually started out as a genomics company with a focus in livestock cloning. So we really started out with cattle and pig cloning. It wasn't until 2005 that we actually added horse cloning. And then, as Malene mentioned earlier, in 2015, we brought in the canine and feline species. In 2018, we had a kind of a buyout from some partners inside the company, and we actually took Viagen Pets and Equine separate from Viagen. So we actually split off the Viagen Pets and Equine side. So now Viagen Pets and Equine focuses on cats, dogs, horses, and endangered species. We've actually set up numerous labs across the country. We basically have set up labs in China, Argentina. We have labs in Canada, as we mentioned before, New York, Brazil. Through the years, we invested over $100 million in research in cloning to become the world's best in the cloning technology.
Wow, you've certainly, I mean, brought that technology forward. £100 million. It puts my research budget to shame, really. I think I'm a long way behind that. But how have you seen the sort of technologies evolve over the years, Sean? Because you know what's great to see Viagen. You've all been, sounds like you've all been there virtually from the beginning, or for a long time. So you must have seen a huge change in technologies over the years.
Yes, I mean, we've seen a lot. Whenever they originally talked about Dolly, they talked about, you know, transferring hundreds and hundreds of embryos to get animals. We're down to a fraction of that. There's a lot of issues with early neonatal health whenever the clones were born early on. And so we got rid of a lot of that. So we've seen a lot of progress, both through the health of our offspring when they're born and actually what it takes to actually make a clone. Right now, we've actually cloned 11 different species, with more coming in the endangered species realm right now. And so we probably we're up in the tens of thousands probably of clones throughout the years, throughout the 20 years. And so a lot of animals on the ground to look at.
And the Brzezowski horse this year, that must, or I think it was born on the 6th of August, wasn't it, last year, I think? That must have been a huge milestone for you. I mean, showing your techniques now to stop breeds from going extinct, that must give you such a sense of achievement, really.
Yeah, it was a lot of fun because, I mean, it was a great collaboration. It was a collaboration between Revive and Restore. They're actually a leading wildlife organization promoting incorporation of biotechnologies into standard conservation practices. And so it's a really neat company, the San Diego Zoo. And so everybody knows about the San Diego Zoo. And so that was actually a lot of fun. For a little bit of background, I guess, about the Przewalski's horse was that they're actually native to Central Asia. And right now there's only 2,000 of them around, and actually all of those came from 12 founder individuals. And so it's a very genetically bottlenecked population. And so it's actually was great for us to be able to use that technology to bring that lost genetics back into the herd. And then that animal, Kurt, who was actually named Kurt after Dr. Kurt Bernstein, who's actually the founder of the San Diego Zoo, basically their frozen ark, which is basically samples that have been saved throughout the years of endangered species for this type of technology to be able to bring back those lost genetics. And so hopefully we'll be able to open up the genetic bottleneck a little bit on that.
And I think we've got a video, haven't we? Is that right?
Yeah, so here you can see Kurt running around. He's quite a rambunctious foal here.
He's got a fair jump on him as well.
He does. He has some energy in him.
Crumbs, that must have been an absolutely amazing moment. And to me, this is obviously proof of concept that this technology can really help species from going extinct. Sometimes, you know, people don't always see the good things in, you know, they might paint a bit of a bad picture on what cloning, things like that. But this is such a fantastic story right the way through. And, you know, as we see more about this, these are just perfectly normal animals now that you're producing. And what other work have you done on the preservation side as well?
So we're also working with the U.S. Fish and Wildlife Service division of the U.S. federal government. Again, Revive and Restore in the San Diego Zoo on a black-footed ferret project. So same principle is to bring back the lost genetics. San Diego Zoo has some genetic samples saved back throughout the time. And so what Viagen is using, we're actually using domestic ferrets as oocyte donors and also as surrogates for them. And so here you can see our group. Our group is Dennis, Anita, Jill, and Carrie. And so this is our group that's working with the domestic ferrets. They actually also do our canine and feline work. They're our New York group. And so what Viagen's doing is using these domestic ferrets as, like I said, oocyte donors and as surrogates. And so it's quite an exciting project. Again, there's only 370 of these ferrets living in the wild right now. They have been highly genetically bottlenecked. They actually were thought twice to be extinct. And so this is a great project. We can bring back some of those lost genetics, open up that bottleneck, hopefully allow these species a chance to survive and thrive and get reintroduced back into the wild, because these animals have been tried to be reintroduced into the wild, and it's actually because of sylvatic plague and canine parvo, they've actually died back out. And so Revive and Restore has a really neat vision, and that is actually to use biotechnology to maybe even help make these animals resistant to those diseases so they can actually come back and thrive as a population on their own without having support from us to basically keep that population going.
Yeah, I mean, we've all heard of the white rhino where there's 2 left in the world. We all hear about the elephants declining and all these species, but we don't think of a ferret and being rare. But the black-footed ferret is exceptionally rare, if I'm understanding. I've got a certain affinity to ferrets. I used to have ferrets as a kid. My first one was called Jenny. She was a little polecat ferret, and that's the only way to get me to school was I used to take the ferret to school. So that's my little story about ferrets. I used to absolutely love ferrets. So yes, but one day she did escape, and we got a bit of a trouble then because she ran around the whole school. Anyway, that's another story about ferrets, but they're exceptionally rare. So that's great to see that sort of work being carried out and showing that it can work. Because obviously that links back to the charity we've done, Nature Safe, and just showing that this work we can do preserving these cells, it's not just about cloning, there's lots of other things we can do with obviously with the cells, preserving testicular tissue down, ovarian tissue down, and so on. But can you explain a little about the process? So you've got the sample now from Elaine. It comes up. Sanaz, and it arrives. So how does the process work, Sean?
Well, actually, I think we have a couple quick videos of our lab. So we just wanted to show a couple videos at the lab since we weren't able to go there. And so I think we have 4 real quick. We'll go through those and then. So this would just be our cell culture lab. And this just basically shows actually taking the samples out. So once Sanaz would actually send us the samples, we would then store them in a doer similar to what Sanaz has. And so it would just be stored in that. So I think the second clip will just kind of show give you an overview of what our cell culture lab is. It's not near as elaborate as Sanaz's because we really only need to break our cells out and take and work with them for the cloning process. So our our lab is actually fairly straightforward here and for the cell culture lab. You can see we have incubators, microscope, and then a biological safety hood, just like Sanaz had in her lab.
Then what's you doing with the process now?
So this would actually be breaking the cells from freezing and actually put them into plate to get them prepared for cloning. And so if we go on to the next, so this would be our processing part. So whenever Sanaz would take the sample, like she said, she would then send them to us whenever a client is ready to clone. We'd break the cells out into the cell culture lab, and then we would take and grow those cells until we're ready for cloning. Once we're ready for cloning, we actually take ovaries from horses. So we'll go through horses. So this is our horse lab. So we'll take ovaries from horses and we'll collect oocytes from those. And so for actually collection, we can either aspirate ovaries or we can actually collect from basically from animals collected from the abattoir. And so we take those ovaries, scrape follicles, and actually take the oocytes out of those follicles. And so this is just, like I said, this is just our cloning scope. This would be a fairly standard manipulation scope. We use a size scope. You can see there's little controllers on the side, right to the left-hand side of my hand that would control in and out. And so that would just be a standard cloning scope. So I'll go back to the process a little bit. So like I said, I just wanted to give you a little bit of an inside view of the lab. So what we'll do is we'll take and collect the oocytes from the ovaries. And so like I said, we can either aspirate those from mares or collect from ovaries themselves. And so basically, whenever we take and collect those ovaries, we would then take and put them into maturation. The maturation media is just we'll take the oocytes from the ovaries and put those in maturation. The oocytes are the same thing as the eggs. And so the maturation media is actually a growth media. And so it allows those eggs or oocytes to mature to a stage at which they're usable for cloning. That stage would be considered a metaphase 2, and it's usually 24 hours after the start of maturation. What we would then do is we would take those eggs or oocytes and we remove the cells from the outside of those, and then we actually put those on the microscope that you would just have seen. On that microscope, we would do what's called a nucleation. So I think we have a nucleation and reconstruction video. If we could show the nucleation video, that would be great, please. So what you're seeing here is just the enucleation of an oocyte. And so basically the pipette there is approximately 20 microns. And so that will give you a little bit of size. The oocyte is typically 100 to 120 microns. And so you see it's going in and you'll see a flash and you'll see just a little flash of light within the oocyte. And that's actually the DNA inside the oocyte. And so if you could loop that one more time, please. And so that's the polar body. So that's being removed. That would be extruded. The first thing that's extruded, so that's DNA. So you'll see the flash. It's really hard to see on this video, but you should be seeing a flashlight inside, which would be the metaphase plate or the DNA, which is inside the oocyte that is just then aspirated out. And so that oocyte now is devoid of the DNA that would have told it to be whatever animal was destined to be. And so then we would go into our reconstruction. So this is where the cells from Sanaz would come into play. So you see the two little circles in the pipette. That's actually the fibroblast from the animal that we want to clone. And so that's basically loaded up into the pipette and then injected back into the oocyte that basically we have removed the DNA out of. And so what we would then do is we would then give that oocyte with cell a electrical pulse, and that causes a fusion process. And that fusion process allows the membrane between the cell and the oocyte to fuse, and then it dumps the DNA into that oocyte. So now that oocyte contains all the genetic material you need to make a new animal. That oocyte reconstruct, we would call it, would be activated, and then it goes on to divide. And so it would go into 2, 4, 8 cells, just like a normal embryo in the incubator. For equine, what we would do is we would then freeze those embryos back at day 7 at a blastocyst stage. Then we can ship those around the world to our embryo transfer providers. They're transferred, well, they're thawed and then transferred as standard embryo transfer. The mares are then basically, once deemed pregnant, are treated like standard pregnant mares. They go on and deliver a natural, healthy offspring. And so that's pretty much the overview of the cloning process.
That's absolutely amazing. I could have to admit, I feel like I could watch. I have to admit, I haven't seen that video yet. Absolutely fascinating how you make it look so simple. I have to admit, I'm sure it's a lot more complicated than you're actually explaining it, obviously. But it looks absolutely fascinating. And typically, is there a few embryos that you do each time, or?
So typically in a given day, we may run, it varies depending on what we get, but it may run through 20 to 40 oocytes that we would manipulate in a given day.
Yeah, right, okay. Now it's a quick thing.
Yeah, there's a question actually quite a few people are asking, how much does the process cost from start to finish?
So I don't know, should we go ahead and let Melaine address the cost of all the species, or you just want to hit equine right now, or?
I think we've got dogs, cats, horses. I think a few people want to know.
Okay, so I can address that. So the first step that we talked about, preserving the cells from the animal, the genetic preservation, and that is $1,600 US. And then there's an annual storage fee of $150 per year. The cloning price varies per species. So our equine cloning is $85,000, canine cloning is $50,000, and feline cloning is $35,000. The cloning prices, you're guaranteed a live, healthy animal. The cloning fee is split into 2 payments. So the client would pay half of the cloning fee as a deposit when we begin the cloning process, and then the second half of that fee would be due when the animal is ready to go home.
I think the exchange rate's pretty good between us and the USA at the moment, so I think that price is quite favourable for us at the moment. And then what do you charge just for storing a tissue sample as opposed to the cell lines?
So yes, we do express tissue banking, which is just the biopsies are taken the same way, but then when we receive those tissues, they're just simply cryopreserved. So that is a less expensive option. That's $500 with an annual storage fee of $100 per year.
Yeah, so I think we charge, what is it, £400, isn't it? So yeah, we're obviously, I think we're very similar to you on that side, and we find that quite popular in the UK. But now we're starting to grow the cell lines a little bit more. So it's not very expensive really, just to store cells down really for that first stage, and then obviously going on to cell lines, is it? And that's like you say, that's across a lot of different species, obviously, on that side of it.
That's right. And also one thing to note is all of those initial fees, your initial preservation fee, and all of your annual storage fees, those actually get applied toward the cost of cloning. So we've had some clients that have stored their pet's DNA for almost 20 years. And so all those storage fees that they paid over the years got applied toward the cloning fee.
Wow. Oh, right. So after about 100 years, they virtually get a free clone in theory if they're there for long enough. Right. So was there any more, Sean? Was there any more to say on your side, or are you you nearly done?
Sorry. Just that we're really excited about the collaboration with Gemini Genetics and with the SAFE organization to help expanding the endangered species work. And so it gives greater access to our clients internationally, and it provides us with great partners, and we're really excited about that collaboration. Yeah,
I quite agree, and we love the work, and I know we keep coming back to it, but God, your work, there are 25,000 species that could well go extinct in the next 3 years. There's nearly 1 million really endangered, and this is one way we can stop species from going extinct. It's quite amazing, and I think we're just at the starting side of it, and the way that you're progressing forward with all your new technology. So I know some of it you can't talk about, but some of it, the way you're moving for all that investment of hundreds of millions of pounds, is really pushing the boundaries, and we can actually maybe be able to use these technologies, you know, with these tissue samples, not necessarily today, but in 20 or 30 years time, there'll be new technologies out there. How can you sort of see it evolving over the years, and what sort of new technology do you think around the corner, Sean?
You know, I guess your imagination is the only limit, is what I've learned in science, and that is, if you can dream it, it will likely happen. So I think, you know, that even from just taking a skin sample to turning that skin sample into basically an animal without having to use an oocyte, you know, you got the iPSC cells, you have so much technology. And like I said, it's just been amazing throughout the years that I think really your biggest downfall would be if you limit it to what you think you have available today, and not allow your imagination to go, because there is a lot of ways to go with this. I'm totally with you. I remember when I started this 20 or 30 years ago, I never thought we'd be doing this. I never thought we'd have a sexed foal born by sex, or a sex semen foal born last year, a Suffolk punch. I never thought that was going to happen. I never thought we'd be involved in tissue banking for cloning. I never thought we'd actually starting off one of the first ever living tissue banks, you know, in Europe. And so it's great to be able to use these technologies right across the board. Do you want to pour a bit in there, Lucy? So we've got another poll question. So really, I think that's a big thank you, Sean. I think that's great to see those videos. I've never seen them before, and it's great. I mean, honestly, I take my hat to the work you do it as if you look like you do it in your sleep. But obviously, it's highly complicated. The stuff you're using is really cutting edge. So just thank you very much for participating in tonight. So yes, I think just got a poll question coming up now, if we can. We'd just really like to hear from yourselves. Would you be interested in actually having a genetic preservation and maybe obviously for either just a tissue bank or going to cell lines, or would you be interested in cloning? So just would you want it preserved, or you're not, you know, you're just not interested? So you're just interested obviously on that poll from that side of it. So just going back, so before, I think Rachel, can you put up the logo of Nature Safe? So I say we're going to talk about Nature Safe. Nature Safe saves saving animals from extinction. I think we might as well just show a bit in here. Do you think? Then we can go back to Blake as well. Then, so just in our lab here, I better just get your goggles on and stuff, and then Lucy can. I'll just show you in here while we've got Nature Safe up. Oh, actually you can't. So you better take that down. Actually, so yeah, I'll be going in the wrong order if I do that, won't I, Lucy? So actually, I think we will leave that for a second. Let's go back to Blake now. In your
questions.
Oh, more questions. Yeah.
And poll results are in.
Poll results are in. So wow. Well, that's. I have to admit, I didn't know what sort of results we were going to get tonight. But Blake, you'll be pleased that 72% of the people tonight said they're interested in doing preservation. So I think that's just shows how many people are really interested in that and maybe getting involved. And obviously, all our details, you can get hold of us or Viagen. So if we can move back to Blake. Are you there, Blake?
Yes, sir.
So that was a great poll that you just said. 72% are interested and maybe in the preservation side of it, which is great really to hear, isn't it?
Yeah, I don't think that could all be just Viagen Pets, friends, and family, right? There's got to be some additional people.
Exactly. There's more than about 5 people watching. I think there's hundreds and hundreds of people. So, yeah, just explain a bit. I think it'd be quite nice. Can we see a tour around your facilities now? This is where all the foals are born. Is that right?
Well, it's not. So it's really important to know that, like Sean's work, for example, those embryos then are cryopreserved. We get a USDA permit for those to come back into the United States. And then we do transfers on those embryos, both in Canada and in the US. And so we have about 5 foaling locations. I happen to be one of them, and I'm actually one of the smaller ones. But I have an air-conditioned foaling barn, which is handy in the summers because it can get really hot in Texas. So I just thought I'd kind of see if I could get some foals to cooperate. You know, for those of you that are watching, there's really nothing highly unique about this facility other than the foals that are here. I mean, it's amazing the diversity that we have on this property. So here I am in the middle of what people think of as quarter horse country, but I've got Arabians, polo horses, warm bloods. Let me think, what else have I got cool here? Yeah, I mean, it's crazy that, well, here's another quarter horse clone that was just born the other day. Let's see, he's taking a nap. Hey buddy, you want to talk to the crowd? And so anyway, it's really, you know, it's remarkable as you think about, you know, the types of people that participate in this. So, you know, it's not just the technology for the rich and the curious anymore. You know, at $85,000 for a cloned horse, it's still expensive. You want to invest only in really elite genetics. But, you know, you've seen here this mare has been allowed to make a continued contribution of her genetic line, which wouldn't have been possible otherwise. You know, these babies here will, you know, some of these will actually go to the Middle East. Some of these babies are just going to go right down the road to clients here in the area. As I kind of step outside here and I try to look over my shoulder, I'm trying to keep internet access here without losing, but there's a couple of warm bloods there that'll be going to Europe. There's another Arabian. I mean, it's just remarkable, the diversity when you think about gated horses, polo horses, warm bloods, and the numbers of people that are, you know, experiencing and benefiting from this technology. So I just wanted to kind of open that up. Maybe it comes up with questions later to kind of see the different things that are going on. As Sean said, really from the point of embryo transfer forward, this looks and feels much like the technologies that all of us are familiar with embryo transfer. We know the value of these foals when they're born, so we obviously do a few special things here to make sure that each one of those foals gets a great start, gets the right opportunity, et cetera. But really, one of the key pieces of progress that we've made in this technology is the quality of these foals when they're born. So we don't have to do a lot of special things with these foals to get them off to a good start. And to answer a question that I know is coming is, tell us about the health and well-being of these cloned foals. And our experience has been over the last many, many years is a healthy foal is a healthy foal. They go on and they live very normal, active, healthy lives. There was a question earlier about the size question, and I think one of the big lessons that we've learned over the years is the importance of the size of the recipient. So, you know, you have to match the surrogate, the uterine capacity for these mares have to match the birth size of the foal. And so we certainly have done a much better, you can see a warm blood mare here in the background. You saw some draft crosses out there with foals on their side. We certainly are doing a much better job on the husbandry side of that. So, you know, in the cases where you might see one that's small, that might have been part of our learning curve early on, where we were learning how important it was to properly match the size of the foal with the size of the surrogate.
No, that's brilliant. It's lovely to look around there and suddenly see your new foals that have just been born, Blake. Well, thank you very much for that. We've got a few more questions, but I appreciate we're actually, as always with all these webinars, we seem to be running over. We've had one person say, "Can we clone Tullis?" Is that right?
No, I think we don't need any more.
That wasn't you, Shirley, was it? Yeah, no, you don't want another one of me. That's for certain. But no, I think what we'll do is, was there anything else you want to add, or can we carry on in our lab here and then we'll come back to you again upstairs, Blake? Or is there anything else you want to show us in your barn?
Yes, that sounds perfect. And I'll stay connected so that we can answer questions. We can show any additional pictures anyone wants. And again, we'll stay hooked until the end.
So with Lucy's now, I just wanted to put out these, we've got our new green bio gloves, now biodegradable. So the company that gave, we did say we'd give them a bit of a plug on that side of it, because they're biodegradable gloves. That's why they're green. So that's our new thing we've done for the new year, is having these new gloves. But anyway, Lucy's going to explain a bit about what we do in the lab here and about the samples. So Lucy, over to you. This is from Gemini Genetics.
Yeah, so hi, yes, I'm just going to explain what we can offer here at Gemini. Our situation is slightly different in the UK in that samples can only be taken in the UK after the animal's passed away. So Melaine was talking about it at the start of this about samples coming from live animals. We can't do that here, they have to be taken after the animal's passed away. So one of the key benefits of our premises here is that we're based in the UK, so you can ship samples to us quite quickly, and especially if you're a UK resident. If you're in Europe as well, you don't have the same restrictions as you do shipping to the US. So if you have an animal that's passed away and you really need to get the samples to a preservation centre, we can help dramatically with that here. And then our partnership with Viagen means that you have the access to the cloning technology should you wish to do so. So in terms of shipping a sample to us, it's similar preservation kit to that you saw with Viagen. So the samples are sent chilled, and one of the key things that I wanted to say is it's really important, if you are considering genetic preservation, that you plan ahead and you tell your vet, because we're dealing with situations after the animal's passed away. Standardly here, the bodies will normally go into frozen storage, and that dramatically reduces your chance of viable sample recovery. So it's just really important, if you're considering genetic preservation, that you tell your vet and you ask for chilled storage of the body after your animal's passed away. The samples can then either be sent when they perform the tissue biopsies in saline solution, in pots, or in sterile bags. We offer a designated courier service here. So if you're based in the UK, we can organise a direct driver to come down and collect the tissue samples for you. That means that they can come to us as rapidly as possible after the animal's passed away. If you're based elsewhere in Europe or so on, we can assist as much as possible in getting the samples to us as quickly as we can and in the safest way. For equines, this is an equine example. We have a full instruction booklet which we can send out to you in advance so that you know exactly what's got to be done at the time you're going to be needing this service. The actual service that we perform here in the UK is the genetic preservation. So this is where we preserve the tissue to prevent them degrading, and so preserve that DNA. We are in the process of moving on to the next stage of cell culture. But at all times, the most time-critical stage is getting the DNA preserved. You don't have any pressure to move on to any further stage than that. We can literally just take the tissue sample and keep it in storage here with you.
Yeah. Is there anything else you want to show, or we showed all this already?
I think we pretty much showed most things already. The main things which Sanaz went through are the safe working, the biological safety cabinet. It's really important because, again, we're dealing with animals that have passed away, that you can be assured that the samples are being processed in the best possible way. So it's really key, because we won't get the opportunity to have those samples from those animals again, that we have sterile working environments. And we follow the same procedures as Viagen Pets and Equine. So on a quality control point of view, you can be assured as well that when a sample comes to us, we will be doing our utmost to ensure that those genetics are preserved for you. And Tullis is going to show you the way we preserve them is in the small vials that you saw Sanaz holding up before. And we actually use to ensure that your sample is safely stored with us and to minimise the risk of any misidentification of that sample. We have an adapted version of the human IVF labelling system called, it's a matcher labelling system, and it produces for each animal a huge number of labels which are all barcoded, which then go onto the small vials that we store the tissue in. And then it also means that we have labels for when we bring those animals tissue for the next stage of cell culture and any other processes. We can use a repeat of those labels to ensure that at each stage we can correctly identify that animal as being that said animal that was meant to be in there. And so Tullis is just going to show you what the cryovials look like in frozen state.
These are the, I just want a big shout out to everybody. God, we've had people from obviously America, Nigeria. Big thumbs. Thanks very much for watching. Iraq, Iran, Pakistan, South Africa, Mexico, Australia, New Zealand. Just incredible tonight. We literally, I think we're touching all the globes all over the world. So please keep in touch with us, everybody out there. We, you know, anything we can do to help on preservation, semen preservation. We love setting up labs, anything like that. Please, please get in touch with us. Rachel's sending out. But I'm going to put another plea. We've already had somebody give a really big donation tonight to Nature's Safe. And in here, this is what can stop animals from going extinct. In these cryovials, we've got some rhinos, we've got a whole different type of species, cryo-reserved, ready to regenerate, stop these species going extinct. So some of the people that put tonight, you know, even if it's a 5 or a 10, I promise you, every single penny will go towards preserving animals from going extinct. So please, if you can do that, I'd really appreciate it. Pushing the word out for Nature's Safe. But anyway, I think we're going to go upstairs now. It's running on, as always, really, really late here. We always get carried away. So many questions, so we'll be able to answer some more. So Rachel, we've got 30-second video, and then we're going to show you a presentation all about the other technology, sexing semen, epididymal sperm extraction, a bit about Nature's Safe. Anything else?
It's also going to be about the further applications of genetic preservation and cloning to the performance industry and rare breed preservation as well.
I promise you there, if you sit, go and grab another glass of wine. You've got 30 seconds to grab that. We're still here going, so my beer's on ice at the moment. So yeah, Shirley Light, get another, top up that bottle of wine, that glass, and we'll see you in about 30 seconds. So, oh, and Dave, no, you can't either. But your request that you had earlier. So we'll see you in a minute, just upstairs. So if you can put that video off, brilliant.
Chloe's the sweetest dog in the world. She goes to work with me. She works out with me. The vet told us she was getting older and I just didn't want to lose her. So my husband got me a Christmas present, Chloe's genetic twin. This is Zoe. We worked with Viagen Pets and they couldn't have been better. They were always responsive, always caring. I love Zoe so much and I'm glad to know my bond with Chloe will continue.
Contact us today to learn how you can preserve your pet. Viagen Pets, love that lasts forever.
Right, so we're back upstairs. Oh no, shouldn't have done that. That's a false start. We've just got to do a screen share to start with, making sure we get it on the right one. Rachel, can you just give me a thumbs up that you can hear me? Well, not a thumbs up, but a yes.
Yes, it's fine.
Brilliant. Right, well, I'm going to hand over, we're going to split this in 2 halves. So Lucy's going to carry out the first bit, and then I'll do the tail end. Lucy does all the complicated bit, all about the freezing, and I can just wrap it up about Nature's Safe. So yeah, over to you, Lucy.
Thank you. Yes, so this presentation, I was just going to give you an overview of some of the applications of genetic preservation and cloning to the main sectors being performance animal breeding and rare breed preservation. Then Tullis is going to take over and he's going to talk about other population supporting and breeding technologies that we have available to us, their application to endangered species preservation, and then close on the charity just launched Nature's Safe. So genetic preservation, as we've already seen in below and so on, is the preservation of DNA for future use. The work that we here at Gemini and Viagen Pets and Equine, the work that we do is unique in that it preserves the DNA in such a way that the DNA can replicate in the future. In terms of biobanks for species conservation, there's several that do store DNA, but it's not in a way that the DNA can be regenerated in the future. So biobanks such as the one here for Nature Safe and the ones that Viagen work with, they're really quite unique in being able to regenerate that DNA in the future. So as Sean went over, the process of cloning and genetic preservation, once we've preserved the genetics, you can then go on to cloning, and it's although the process is very simple, the actual theory, the process is very complicated, sorry, the actual theory behind it is quite relatively simple in that we're taking the DNA of the animal we want to clone, inserting that into a host egg cell, and then that goes into a recipient mare that can then carry the animal to full term. And really, from what we're actually looking at when we're doing genetic preservation, we're actually looking at the animal that we see in front of us as a mixture of the genetics of that animal and the effect of the environment upon that animal. And what we're looking at in genetic preservation and cloning is really capturing, and it's also true when we're just breeding animals standardly, we're looking at capturing the genetic aspects of the performance that we see. So whether that's jumping ability, dressage, eventing, or in your companion animals, if you're a gun dog, for instance, your traits of performance there. And that's what cloning is really comes into place, is where you've got an animal of strong, desirable genetics, you're wanting to capture those exact genetics of which you'll never have access to again and regenerate them in future generations. For performance applications, it's obviously best suited to traits of higher heritability, which is also true of standard breeding. You're looking at carrying on traits which are passable to future generations. We've got here horses obviously shown, we've got sniffer dogs, working dogs, guide dogs, sectors such as that. So the main applications I was going to discuss in this presentation are performance animal breeding, rare breed preservation. There is also obviously importantly another application is the emotional preservation and regeneration of a personally valued animal. So not everyone will have a performance animal, but they may have an animal that's really, really special to them. And there's nothing really wrong in them wanting to keep hold of a lifelong bond with that animal and bring back that animal that's very special to them. So there is a third aspect there at play. So for performance animal breeding, I was just going to touch on a stallion that's actually resident here with us at Stallion AI Services. He's the one in the lower picture here, Merker's Gem, and he is a clone of a gelding Gem Twist. There were two clones produced from this gelding. The other one is Gemini, and he stands in Europe, and he's in the top picture there. Sorry, Tullis has took my mouse. There we go. So what we're looking at here is Merker's Gem has essentially a known genetic value. He's a clone of a horse that's already been there and been out there and done it. We know that there is the same genetic potential in Merker's Gem as there was in the original Gem Twist. Merker's Gem therefore has the same genetic potential to achieve the same performance as the original Gem Twist. And so in doing so, Merker's Gem enables people to breed from him as if they were breeding from that original horse. And it's that ability to be able to capture those genetics and transfer those exact ones to future generations, which is one of the key applications here. There's also the question that we may be able to have improved performance of the cloned animals to the original, because we could factor in something such as environmental optimization. Having had the original with us and known what worked for that original animal, we can then apply those factors to the cloned individual, allowing them to reach more of their genetic potential that may have been limited in the original animal because we were doing a lot of trial and error essentially in training and so on. Other aspects for the performance industry are breeding from castrated males and neutered females in the non-equine sense there, obviously. So in the picture here, we have the legendary Milton. He was a gelding. He was a very, very good competition horse though, but because he was a gelding, he has no progeny. So cloning enables us to generate offspring from such cases. Also increased female contribution. Mares especially, this is more so for the equine industry where mares are limited with a long gestation period and only one foal every year really maximum. So cloning would enable you to increase female contribution to breeding. There's also then continued influence of desirable genetics beyond the lifespan of the original horse. So we can preserve a tissue sample, keep it for a very, very long time beyond the original's lifespan, and then bring back those genetics in the future, when we may, for instance, have bred towards one characteristic and then be wanting to breed back in a certain characteristic that we've lost. And that goes into my last point there, recall of previously lost genetics and recall of genetic diversity. So I think it's really important in all sectors. When we're thinking about breeding, we'll go on to rare breeds next, but in all sectors we need to be aware and think about maintaining our genetic diversity when we're breeding. And this is another area where cloning can help. So next application is rare breed preservation, and this is actually the founding reason behind Gemini Genetics. Tullis was at a rare breed conservation meeting when he met Blake Russell of Viagen Pets and Equine, and it was on that meeting and being able and wanting to do something to help our rare breeds, who a lot of them are really battling against the clock to save their breeds. It was that really brought the founding behind Gemini Genetics, because by being able to preserve a tissue sample, we can capture the genetics that are currently available in populations and preserve them down. Extinction is very much a real prospect for some of our rare breed equines. There's 14 native to the UK, 12 are considered rare, 5 of which are critically endangered, and their populations are shrinking all the time. So using tissue banking, genetic preservation to capture genetics that are currently available in declining populations is really important. So a lot of you will see that in a lot of Tullis's presentations, he talks about the extinction vortex and how really things that we've not known are important. For example, when we started breeding, we've not considered the implications of inbreeding and so on. We've also got loss of use of several of our, or declining use of our native breeds, economic pressures and so on, that have led to less reproduction rates, smaller fragmented populations. And so we carry on this decline where they've got really shrinking genetic pools until we face extinction, of which that's quite a real prospect for many of our natives. So genetic preservation provides an opportunity to bank current genetic material. With each animal that passes, you won't get those genetics available to them again. Yes, you can do semen preservation, but that's not the exact genetic profile of that individual. Also, when we're thinking of preserving mare lines, oocyte-free egg freezing for mares in equines in particular isn't very successful. So this is a really good method of preserving female lines in particular. And the project that Viagen did, the birth of the Porolsky horse, it really demonstrates how cloning can be used in declining populations to bring back lost genetics and really help increase genetic diversity. So this is obviously, the Porolsky horse is a closed population. It's declining. There's been a lot of inbreeding in the past, and there's only captive populations available. So being able to bring back a sample from 40 years ago has really brought an immense amount of genetic diversity back into that population, which otherwise wouldn't have been able to do so. In terms of the samples that you send to us, like I said before, the ones that are coming to us are slightly different in that they're after the animal has died. So we ask for sample sites from several areas so that we can freeze down numerous sample sites for you to help increase sample viability and the security of genetic preservation. So you've got neck, chest, abdomen, inner thigh, dock. We typically ask for four different sample sites. Ear tissue for us is one that we often request because these animals are, because the samples are mainly coming to us after the animals died. And ear tissue tends to be one of the more viable sites as well. So we do ask that to be included where possible. Developing sample types and ones for the future, blood, urine and hair. That would obviously increase blood and urine, for example. We could take samples from animals while they're still alive using them, and it increases availability for people. I touched on this below, but just to repeat it again, the samples that we have here, we identify them with a really safe and secure matcher labelling system adapted from the human IVF world. And it really provides a high security of samples and safe and secure identification of those, so we can be sure of the correct identification of the many tissue samples that are in storage with us. Moving on to future applications, just wanted to touch on, and Tullis mentioned before, it's not just all about cloning and preserving these samples down. In the future, we may be able to use pluripotent stem cell technology. So this will be where we, during the culture of the cells, we try and manipulate them and reprogram them to develop into different cell types. So for example, we could reprogram them to develop into ovarian and testicular tissue, or sperm and egg cells to enable the generation of reproductive cells. And so we're not cloning directly, instead we're producing reproductive cells from the stored tissue. And this is a human example here of pluripotent stem cell technology. It's not yet available in the animal sector, but that's just a future use that we may be looking at doing. And then it's
Right.
handing over to Tullis for his bit.
Well, how do I follow that? Yeah, well, I mean, it's great to see obviously this other attributes to taking skin samples and what other things we can do with them. But I'm just going to go through some of the other preservation techniques. We said about preservation and cloning, but there's lots of other things that we can be doing to preserve these. I know some of you may have seen this before, but I'm going to show you again anyway. So here we go. So one of the things that we do for quite some time is post-castration epididymal sperm extraction. That's a mouthful, even if it is late at night. So this is where we actually, from an animal that's just been castrated or recently deceased, as long as they get the testicles to us, really within 24 hours, we can actually take off the epididymis, and we can actually harvest the semen off that. And we've done this from over 150 equines now, and it works very, very well most of the time, unless the animal's had a certain disease or it's been chronically ill for a certain period of time. And we started doing this on all other species. So if ever an animal dies at Chester Zoo at a sexually mature age, and it's got they send us the testicles, we can often get cell semen from that. So on the rare breed side, you know, this was having a castration, and we actually managed to get 18 doses of this rare breed fell pony when it was being castrated. So we then looked into semen harvesting of some other different species. It does open up a lot more complications, the size of the testicles and so on. Have we got pictures of these ones or not tonight, Lucy? No.
I don't think.
No. So in other words, from a horse, it's quite easy. But you try and take semen off from a mouse deer's testicles. I mean, they weigh barely a gram. So, but we have managed to do that. We have managed to freeze it down. We need to find out what cryoprotection to use. But I find that side absolutely fascinating, and it's a really interesting work because you've got to think outside the box when you're presented with this with these species, because some of the time they've never been done before. So that's the epididymal side. We're moving on to the sex semen side. This is where we can we use a lab up the road called Cogent, and they can take off. There's about 4% more DNA in the X-bearing sperm, the female sperm, than the male sperm. So there's about 4% more DNA in there, and you can home in onto that DNA and put a fluorescent stain in there. That means it shines brighter, those ones with a bit more DNA. And you can get a laser, and it can give a positive or negative charge to those slightly brighter ones. You've got two magnets, and literally the positives, the opposites attract, so the positives will go in a negative pot, and so on. And it's well, it's virtually over 95% accurate, so it works very, very well. And we're just starting to look into the frozen side of this now for ICSI. So this is this next video is a Suffolk foal. There's only 72 or 78 females left in the country, 300 in the world, and they need more females on the ground. And this is just a short video of last year's foal. And after 11 months of waiting, Ruby has produced a filly foal. And it's not just any old foal. This is a foal born by sex semen. We believe it's the first ever in the world for a rare breed animal to use sex semen. And using these technologies can really help us breed. There's only about 70 or 80 females left in the country. There's only 300 left in the world. They need all the tools of the industry, and just quite incredible now what we can do with science and technologies. And using this technology, maybe, just maybe, we can help stop these breeds from going extinct. But this holds the future. Little girl here. So it's the first steps. This little girl. So this was a combination of a load of different people all coming together to make this happen. I must admit, I feel like I've got I'm the luckiest person alive. This is it's just great to be able to do stuff like this. I don't know, I just find it absolutely amazing, and it's great to be able to do something for rare breeds, and it really gives you a sense of achievement as well. So, you know, Christopher Price, who's the Chief Executive of Rare Breed Survival Trust, said this project really demonstrates the viability of new technologies to selecting foals to increase the breeding population more rapidly than they would really achieve through normal traditional methods. So yeah, hopefully this will sustain the future. Our next thing is to do frozen sex semen, which we're already doing, but we're just doing this really for the ICSI programs, and this is a way that can be used for in the future. And we've already got sex semen, ICSI from Big Star, and some other ones out in Italy already ready, so people can actually obviously pick it on that side. And we've got some Suffolk Punch sex semen as well. So as we all know, the ICSI is really coming along leaps and bounds now. Other things that we can do is take oocytes. This was down at Beaufort. Emma Thomas, they managed to get 8 oocytes from this mare, and managed to get 3 embryos. It was sent off to Italy, and they got 3 embryos matured and frozen by this mare that sadly passed away. Does this video play? Yep. So this is one of the first ever actual videos taken. It's done on a time-lapse camera. This is a embryo dividing. It's a few years ago this was taken, showing how it was taken a picture every 20 minutes and speeded up. So you can see from when the ICSI's performed, actually this foal starting to, or the cell starting to divide again, obviously at the very early stage of being an embryo. So we've had our first foal. This is at Edward next door through ICSI. And the beauty about this, obviously, especially with the sex side, we can preserve the male and the female side. And obviously on this side also is a much greater chance of preserving the embryo is frozen. They're much easier to freeze at when the ICSI is performed, rather than normal conventional freezing for embryos. So this really brings us on to the rare breed side. And again, there's a lot, I see there is still hundreds and hundreds and hundreds of people watching it. So I really thank you for sticking with us. It's fantastic. I'm just going to, sorry, I know I keep banging you over the head, but please, if you can do a little bit, all our work that we've done on the equines, we've transferred a lot of our knowledge onto to other species as well. And we stand at a pivotal time now of really making sure that these breeds don't go away from us. So I know I've said it a few times. So Nature Safe is something, a charity. We got charitable status literally 4 days before Christmas. And it's the SAFE stat is the acronym Saving Animals from Extinction. It's a living gene bank, and it's really there to bank tissue samples to stop these animals from going extinct, or we can reintroduce these animals. And the fantastic work that Viagen's doing out there, we've got to bank these samples, because if we don't have anything, it's all right for us. We're going to be maybe okay, but our children, our children's children, are certainly going to come into a planet with very, very few animals in. So we've really got to be, I think it's our duty of care really to be able to use these new technologies to assist them. You know, as I say, there's 1 million species at risk. You know, 40% of amphibians, 26% of mammals, and 40% of avian species. And we're in free fall. I know you see it on the news, but it's not fake news. This is actually happening here and now. So extinction happens normally. You know, that's normal evolutionary traits. Extinction happens, but you can see this line going exponentially out of control, and that is where we are now. And what we have to remember, our biodiversity of our planet, us as human beings, is held together by the biodiversity. So in other words, if we lose certain parts of our biodiversity, like the plankton or the bees, we too suffer because we are held together by this very, very fragile biodiversity. And we lose any particular species, it can come tumbling down. So I think it's something we have to be wary of. And I must admit, I've just started to get into this and more, and I find it absolutely fascinating and what we can do to try and reverse that. So Nature's Safe is one of the first living charity banks dedicated to the preservation of biological samples in Europe, where we can store these cells indefinitely. And as I say, we just got the launch. We officially launched on the 18th because the Telegraph was doing a piece on it. Yeah, so it's, as I say, it's preserving for many, many species. The developing techniques of cryopreservation of culture and multiple mediums for multiple taxa to provide a living cell bank for endangered species. That's what it's all about. And we're trying to do this not just in the UK, we'll be spreading this out, hopefully setting up satellite biobanks all over the world with this technologies that we're developing, what Viagen really showing us as well and developing, and from be it skin, be it semen, be it oocytes, be it tissue samples from ovaries or testicles, all this is so important to freeze down. And this is how the process works. So we've seen how the epididymal works. So we can take the sperm from here and then we freeze it down just like we would do normal freezing semen. The only thing we do add something to it to wake the semen up. This is the preservation of testicular tissue. So hopefully in years to come, we can start to produce sperm from these once we freeze it down. And the importance of freezing it down in the cryoprotectant that it can be brought back to life in the future. Ovarian tissue. This is some work being done by one of our trustees, Professor Susanna Williams from Oxford University. She runs the Rhino Fertility Project, and they've been looking about actually getting ovarian tissue and freezing it down, thawing it out, and then being able to produce oocytes from that tissue in a petri dish from the rhinos, which I find absolutely mind-blowing what technology can do now. So we're going on leaps and bounds, and this work is ongoing. And maybe, just maybe, we can preserve some of our really critically endangered species that are out there. Lucy, you better just explain this one. This is above my pay grade. This is how does this one work?
Well, it's the same one as I was saying about earlier with the pluripotent stem cells. So it's collecting a skin sample from an animal, preserving those genetic downs, and then reprogramming those cells so that they turn into different cell types, so that we can, for instance, program them to be able to produce egg or sperm cells. So this again enables us, so we're not just limited to just recreating the original animal that we took the skin sample from. It means we can actually produce reproductive cells from that animal, and so actively use the original preserved skin sample in future reproduction and breeding programs.
And I think that was touched on tonight as well from Sean, wasn't it?
Yeah, we mentioned it a couple of times. It's not currently available, but it's something that definitely as an industry, it's been looked into and researched. So it would be a future progression. So even if you're not interested in preserving genetics for cloning, well, if you're interested in this application, then it's definitely worth getting samples preserved down, and especially with these rare and endangered animals, getting the samples banked now so that we've got them for in the future. So when we have these technologies available, we've got the samples there to be able to use them.
Sorry, yeah, switch on. Sorry, that. And then this is obviously for the cloning process, exactly what Sean and Blake and everyone was talking about before, wasn't it?
Yeah.
By taking an ear sample or skin sample and then being able to take the nucleus out and then put it into an egg of a host animal and activate it, and then obviously stimulate it to grow. And then obviously this is some work we're doing with different species, and this is what we've actually got here from a black rhino that passed away. We got its ear sample and preserved that, and that's sitting in our freezer now for ready for the future. Other work that we've been doing some work out in Africa, working with Imka Loomis and her team at Rhino Force, which is not Rhino, yes, and Cryovolt, and they've been doing a lot of the work with. We collected off 7 bull elephants when we were out there, and it was just amazing work. It's been collecting off these wild elephants. We were providing the semen extender that we use on the equines, and we've used that actually now to freeze down these amazing elephants. There's something like 10 million elephants at the beginning of the century. There's now about 400,000 elephants left, and they reckon by 2025 there's only going to be 200,000 elephants left. You think that's a lot, but it's just shrinking so quickly that a lot of these places there's not many wild elephants left. They're in huge parks, but then you start to get the inbreeding take place. That's why it's important to collect off these animals so these genetics can then be reintroduced. And what Lucy was talking about, the extinction vortex, is happening here and now to wild animals. And actually the extinction vortex is what makes a species go extinct. Yes, it's lack of numbers, but it's not just lack of numbers. It's the inbreeding coefficient going up through the roof, and then everything else falls apart. So if we can reintroduce new strains like Viagen doing the Brzezowski horse, this can actually bolster a breed and keep it going for this. So this is together we can obviously make a big difference with this. So yeah, one last plea. I'm only going to ask this one more time. I know, Deirdre, please, if you can, when you get it, even if you donate £5 or £10, this can go a long way. And half our viewers did it. We can preserve 50 different endangered animals. That's how easy it is to do. And I promise you, every single penny will go nowhere else apart from preserving these animals. So if you can do that, and you'll get the email hopefully tomorrow. So yes, we've got a big giveaway as well. Because we're going to answer a few questions after this. I know it's getting late, so we'll ask just a few. But Rachel, have we got a winner out there that can for this preservation side? Because there's 72% of the people actually wanted it, so hopefully one of those 72% is the lucky winner tonight. Can you hear me, Rachel? You're on mute at the moment. You haven't gone. Oh no, you haven't gone. I was going to say you haven't gone to sleep. It's that late now.
I haven't gone to sleep. No, not at all. How could I? So the lucky winner of the prize tonight is Alison Kenner.
Right. I don't know what country you're in. Hopefully in the UK, because it makes it a little bit easier to get the sample to us. Remember, you've got to get the sample to us, obviously, but yeah, we will obviously freeze that and store that down for you as we go. Just to say, the next webinar, don't go away yet, because we have got more questions, but we're just going to go about the next webinar. It's all about preparing your stallion for the breeding season. It's sponsored by NAF and the new fertility product they're bringing out. We're actually going to do a little bit, not just in the sport horse world, we're actually going to go and do a bit in the thoroughbred world. How do they prepare their stallions, covering all those mares, sometimes 3-4 mares a day. We're going to go behind the scenes and see how they do it as well. So it's going to be, we're going to obviously speak to Bart as we normally do, and Desiree as well, with their years of experience in stallion work, and how do they prepare their stallions. It's so important how you prepare your stallion at the beginning of the season will end up at how good your pregnancy results are through the season. I promise you, it's one not to be missed. And that's in 2 weeks' time today at 7 o'clock, usual time, free of charge again. So we'd love to see you then. But again, we'll send you out some more details about that. We will going to go on to some questions in there, but I am just going to thank all the people from Viagen as well for tonight. They sponsored tonight. They're the ones that obviously made it happen. It costs a lot of money to put these webinars on, and they've come up, and we really appreciate that. And so, and Gemini Genetics has also put its hand in its pocket a little bit as well, I think. So, so I really appreciate that side of it. Also to thank British Breeding, they put these on tonight. So a big thank you to them. And they've got the stallion event, the virtual stallion event as well, on the 1st of February. Yeah, is that right?
No, so they've got...
register before the 1st of February,
so
sorry.
if you register before the 1st of February, you'll receive a free copy of the February issue of the British Breeder magazine, which contains a full stallion catalogue. And they're holding the event virtually on 3 consecutive Wednesdays. So the 10th of February is for eventing, 17th for show jumping, and 24th for dressage.
And if you register before the 1st of February, you will receive a free copy of your February Breeders magazine, which contains all the stallions in the catalogue. Is that right? Right, brilliant. Well, thank you. Don't go away because we have got to ask a few questions. It's just so we can get back to the screen, because I think that's the last slide. So if we stop the sharing now, are we on the full screen now, Rachel?
Yes, you are, yes.
Right. Oh, what's happened here? There we go. So there's a few questions coming. I think one of the questions I saw, Blake, was someone said, why can't they freeze these samples and then send to you? What happens to those cells? Why can't that be done?
Yeah, so it'd be great if, is Sanaz still on? I know that Dr. Walker said he needed to step in and start cloning. But essentially, when you freeze these cells, it's the damage that's done to those cells when they're frozen. And again, that doesn't 100% mean that we cannot make them work. It just reduces the viability of those cells in culture. Lucy, do you want to add to that?
Yeah, I was just going to say, freezing the DNA, with freezing the samples down without having them frozen in the chiroprotectant does exactly what Blake said. It just freezing them down in raw state, there's nothing there to protect the DNA from damage to those when exposed to that very cold temperature. So it's really important that you try and avoid the risk of the samples being frozen down, because it does dramatically reduce the chance of viable sample recovery when it comes to us. So that's what I was saying before. For those of you that aren't able to take the tissue samples from animals while they're alive, it's really important you talk to your vets and just mention you're thinking about genetic preservation. So when your animals pass away, just request chilled storage of the body while you're making arrangement for the samples to come to us for genetic preservation.
And a chap called Chris has asked, can you clone the mammoth?
Well, it's certainly something that lots of people are talking about today. I think the real challenge will be finding complete DNA from the mammoth. And so I think today when they talk about cloning the mammoth, what they're really thinking about is how close can they get to the mammoth, because then they're going to have to find appropriate oocytes, appropriate surrogates. We see all the pictures of, well, that's simple, that's an elephant, but it's never that simple. But I think Sean made a great point earlier, and that is, our imagination is really our only limit. So as we go forward, we're going to learn more. And so the answer to that question is no, not today, but who knows what's possible down the road.
And Faye, I think there's a...
Yeah, we've had a question from Diane. She said, are you doing any gene editing prior to cloning? If we wanted to preserve the genetics of an animal that suffered from a genetic disorder, could this causative gene be edited so that the clone would be free from this disorder?
I'm glad they asked you that one there, Blake, because I'm lost.
Yeah, and that is both a terrific and an important question. So the answer today is no. We do not do any editing to the DNA, and therefore you get a genetic twin to the animal that you submitted. However, obviously the technology is available for those edits to occur, and a number of those defects, they're either single allele or a small number of allele defects that can be corrected. And with current CRISPR technology, et cetera, that can all be done. However, there are regulatory issues around that, which are currently being worked on and better understood. So we have produced a lot of gene edited animals for biotechnology, right? Whether that was for human health work, et cetera. But we do not produce any gene edited animals in our dog, cat, and horse work. But I suspect down the road that animal welfare reasons that there will be terrific opportunities for us to correct some of the defects that we know exist in our populations.
No, that's brilliant. Someone just asked just that basically where else can they donate on a website? Yes, they can. natures-safe.com is where the charity is, a charitable website as well, where you can do that. Sorry, Faye. Yeah.
Yeah, another question come in. Is cloning birds a different technique, or do you use the same method? How's that done?
So Viagen are simply doing non-primate mammals. And so when you get into birds, that is I can't even imagine how to talk that through intelligently. That's something we'll have to hand to Sean or someone else at another time.
Oh, someone's asked, how can you get a Merkish gem foal? Well, you just call us up in the morning and we'll let you know on that. How long can the genetics be preserved before cloning? I think we sort of said this can go on for years, can't it? Is that right?
Yeah, I mean, the oldest samples that I'm directly aware of are about 40 years old, but there was no degradation or loss of cell viability once those were used. So in our view, we've seen no degradation over time. So we suspect that should be able to go on for quite some time. As long as you go back to Lucy's first step, as long as they're cryoprotected and stored properly, then they can have a long, healthy life.
So if you have a sample that's been frozen down and stored, once that has been thawed out, how long is that process then from that starting to the baby, the foal, kitten, puppy being born?
Yeah, so the good news is those people that want to clone a dog or a cat, they've got about a 60 to 65 day gestation period. And so the longest wait is gestation. So in horses, we're looking at something between 325 and 365 days. Actually, one of the foals over here, I nicknamed 370 because I just foaled him the other day and he was 370 days. So he made us wait a while. As Sanaz and Lucy said in the beginning, there's about 2 to 3 weeks involved in the cell culture process. And then just depending on the waiting line at the queue, then there's usually about a couple of weeks involved in getting into the queue and the cloning line. Literally, those embryos are produced in a matter of a day or two. And then in the case of equine embryos, they're allowed to mature and develop for a week, and then frozen back and then shipped to the location where we do the transfer. So it's typically about 1 year on a foal from the time that you submit your sample to the time that you have a healthy foal ready for delivery. We keep the foals with us for up to 60 days. And that's important for a number of reasons. Number one is when it comes time to insure a foal, it's much less expensive to insure a foal at 60 days of age than it is at birth. And so that helps the client. Number two, if there is any issue with the foal, then those things are very well recognized in that first 2 months of time. And then number three, it gives the client time. As I said, gestation length on horses can vary, so they don't know exactly how to prepare the logistics. Well, once the foal is born, then they've got 60 days to prepare for the proper delivery and landing spot for the foal.
Right. And someone asked what the cost was in the UK, and I think it's £400 to preserve it. And someone asked what is the USDA? That's your government vets, is that right?
That's the U.S. Department of Agriculture, and they're the people that control the regulatory side of the import and export of horse tissue and reproductive products.
Yeah, I'm sorry, there's literally we're up to 120 odd questions. We're never going to get around them all. But anyway, Faye, is there?
Leslie asked a question that I saw before. It was about the surrogate mares, or the surrogate that's used, and whether any of their kind of traits will pass on to the foal or whatever, and any kind of how do you block that? Can you block
Yeah,
that?
so the surrogate mare is not providing any genetic contribution to the foal for sure, right? But there are a number of questions around learned behaviors that foals might pick up from their recipients. So in our case, as I mentioned before, we certainly try to make sure we're selecting mares of the appropriate size to carry that foal, but we're also looking for mares that are easy to handle, easy to be around, don't carry a lot of, we're going to try to have mares that only bring the best learned habits to their foals. But our experience has been that these foals, they don't tend to carry on traits from their mother, unless, again, she's just an extremely excitable, nervous, running, and might teach the foal, but we can get around those things too, typically.
Yeah. Okay. Right. I think we're over 2 hours in. I always say we try and wrap it up after 2 hours if we can. We've still got hundreds of people watching. We've had some amazing comments coming through saying, it's been great to see you over there. And to me, I've really enjoyed tonight. It's been a different one than we normally put on. Alison's just wants to say a big thank you to everybody. It's such an amazing experience. So thank you. Deirdre said very interesting. So I've really. And
Tullis, would you just let us, if I can say, if you have any questions, if you go to viagenpets.com and you do the contact Viagen, you can send any question that you may have, and we'll make sure we get those answered right back to you right away. So that's just viagenpets.com. Click on the contact Viagen, and I'd be happy to answer any questions.
Yeah, and I think there's a lot of, we try and get back to everyone's questions. We're up to 100, I say over 120 now. So what we will try and do as much as we can, we will try and get back to as many of these people as we can in the next few weeks. So we might just share that a bit with you as well, Blake, if that's okay. So I really appreciate, we're getting so many great comments and from all over the world. So just one after another. So I really appreciate, Marlene from Wales to say what a fantastic evening. So great, thank you for joining us, and so on. So I better not read them all out. You've got work to do most probably, Blake. So it's quite late for us. It's 9:15 in the evening. I've just got to thank British Breeding again. Rachel, gosh, she works. She's here all night and putting these on. So Rachel, I really appreciate you doing all the background work. I thank Lucy and Faye here tonight, who's Faye's in the background asking the questions, and Lucy's obviously the brains behind all this, sorting all out. And all your team out there, they've been explained it in a way that I think understandable, and we could really sort of get how it all works. So well,
it's an honor to be a part of this. We're thrilled to call you guys our friends. We're thrilled about all the new friends we've made around the world today. So again, thank you for being a part, and let us know if we can ever help.
So don't forget, everybody, see you in 2 weeks' time. It's all about the stallion preparing for the stud season. So it's good night from everybody else. Good night in America. Well, it's in the afternoon for you.
Good afternoon. Yeah.
Yeah.
Okay.
Bye-bye. Again. Goodbye. Many thanks.
Goodbye.
All right.